Good morning, good afternoon, good evening. I'm Carla Rothlin, professor at Yale University and co-founder of Global IMMUNOTALKs. It's been a long time since I had an opportunity to introduce one of our Global Immuno speakers, so I'm absolutely delighted today to introduce Dr. Anne O'Gara. Now, before I do so, please, I would like to invite you to next Global IMMUNOTALK that is going to be given by Dr. Hao. So now back to Anne's introduction. Anne is a principal group leader at the Francis Crick Institute in London. She is original from Gibraltar. She obtained her PhD at the MRC in London in microbial biochemistry, and then transitioned to work on cytokines, which I think have been a passion of her life, and the immune response during her postdoctoral fellowship. After that, she became an independent group leader at DINAC's Research Institute in California. I believe those were transformative years in her career, as many fundamental discoveries were being done at DINAC, some by Anne herself, and I believe Anne will share some of that with us today at the beginning of her talk. It was during that time that Anne recognized the fundamental regulatory functions of IL-10. And after spending about 14 years in the US, she was recruited back to the UK and formed the new division of immune regulation at the National Institute for Medical Research. In 2015, the National Institute for Medical Research became a major part of the CRIC, where she also continued to contribute her leadership skills as an associate research director. As we will learn today from her talk, Anne has extended her research interest to understanding the immune responses in tuberculosis. She has done extraordinary contributions to science that have been recognized in many ways. I would like to mention that Anne was elected as a fellow of the Academy of Sciences UK in 2005 and a fellow of the Royal Society in UK in 2008. Anne is also a member of EMBO since 2009. So Anne, is a true delight to host you today. Thanks so much for accepting our invitation. We very much look forward to your Global IMMUNOTALK entitled Cytokine Interference in the Immune Response to Pathogens with a Focus on Tuberculosis, How Transcriptomics Can Lead the Way. Thank you, Anne, for being with us today.
Thank you, Carla, for that very kind introduction, and also to the organizers for inviting me to this fantastic series of Global IMMUNOTALKs, which I think have revolutionized immunology and delivery of immunology to people all around the world, many who could not access conferences, and hopefully to a lot of young people. So thank you very much.
Thank you so much. And so, as you know, we love to learn a little bit more about our invited speakers. So you have had an absolutely impressive career. So the question that we have for you is, we'd like to know, what is the trait in your personality that helped you the most along this fabulous way?
So, you know, science has its little hurdles, as we all know, but to be honest, what really drove me is my passion for understanding research, for understanding the immune response, its response to infection, the regulation, and all the pathways that control this fantastic process. And I get very excited about data, discuss it with my postdocs, my students, and my collaborators. Sometimes you can spend hours, so we don't realize the time's going by, or maybe I don't realize, they may be exhausted by then. But that really has helped me throughout my career, is this curiosity and passion for science and data, but also seeing students and postdocs develop into their careers, albeit professors, whatever they do in life that they then are happy and succeed in, makes me also very happy.
That is so true. That is so true. Thank you so much. And so I think we're all really looking forward to your talk. If you want, you can share your screen now, and we are all ears. Perfect.
Okay, I even have the pointer. So Today, I chose the title Cytokine Interference in Immune Response to Pathogens with a focus on tuberculosis. And I want to show you what the last years have helped us, how transcriptomics can lead the way. But before that, just to remind you that actually I'm an immunologist. I was trained in cytokine and immunology, immune response, but I started off in microbial biochemistry and came back to the immune response to microbes along the way. So as Carla said, I actually started my independent group at the DNAX Research Institute after doing a short postdoc there. And I was very fortunate to join this small institute in California, buzzing with exciting data. Now it's Merck, and it's actually moved from Palo Alto to South California, southern San Francisco. So what was really exciting is that when the year before I joined there, Tim Mossman and Bob Kaufman, shown here in this photo, for you young people that may not remember, discovered Th1 cells making interferon gamma responsible for macrophage activation and delayed type hypersensitivity at the DNAX Research Institute. And conversely, they found that T helper cell clones conversely could produce IL-4, 5, and much later 13. And this discovery was a seminal discovery for which they were acknowledged with the top citation of biology for 10 years. And these cytokines are the Th2 activated eosinophils and mast cells, while gamma interferon activated macrophages and combating of intracellular pathogens. So then after this discovery, many others went on to show that in addition to these responses, the Th1 and Th2 cells could also control other responses. But at that time, there was a big debate as to do these occur in vivo, and they had isolated them from chronically infected or stimulated mice. One has to remember this. So I was asking this question because I was working on B cells and cytokines I won't go on to now. And Ken Murphy in University of Washington, St. Louis, was asking a similar question. And we asked to collaborate with Ken because he'd made a TCR transgenic mouse, which could recognize a peptide antigen of albumin. So Ken, with Chi-Song Shea, his student, we set out a collaboration with Stephen Macatoni, my postdoc in the lab, and went on to show that actually microbial products, for example, Listeria, LPS, could activate macrophages, provided the T cells were appropriately stimulated the T cell receptor, then cytokines were produced, and IL-12 we found was the dominant factor in driving Th1 development and the production of high levels of interferon gamma, and that turned out to be a differentiation process dictated by transcription factors and signaling pathways that I haven't got time to go into. And also, we found that dendritic cells could also produce IL-12 and thus cause the driving of this Th1 response. So, as I said, after these discoveries, it was later shown that Th1 cells were fundamental by the activation of macrophages in control of Mycobacteria, Leishmania, Salmonella, and other intracellular pathogens. And conversely, the Th2 cells, Bob and others at DNAX had shown eosinophil and mast cell activation, but these also found to be important in anti-helminth responses such as schisto or H. polygyrus later, etc. But the problem with these responses is that too much of them, or in the wrong setting, they could cause inflammatory pathology, such as colitis or allergic inflammation, and thus there had to be a break. And this was way before PD-1 and these other negative regulators. And so at DNAX, we were looking for a negative regulator. And so the discovery of interleukin-10 happened at DNAX. Paulo Vieira was the postdoc who worked with Kevin Moore to clone the mouse and the human IL-10. And this was based on an activity that Tim Mossman and David Fiorentino had described as a cytokine synthesis inhibitory factor. So how did I get into IL-10? Well, I was looking at B cells to see if they produced cytokines or responded to LPS or other triggers differently to macrophages and dendritic cells, to regulate the immune response. And we found that B cells made interleukin-10, and yet they could stimulate Th1 cells, which were originally shown to be inhibited by IL-10 in Tim Mossman's study. And so what we found was that IL-10 inhibits the production of IL-12, TNF, IL-1 from macrophages, and inhibits IL-12 production from dendritic cells, and therefore provided a brake to Th1 development that we in Ken Murphy's lab had determined, and IL-10 could really shut all this off, as well as turning off the activation of NOS and ROS in activated macrophages, was shown by others, and activated by gamma. So IL-10 shut off all these processes. And I should say that René de Valmalfite did similar data in human at DNAX, and so IL-10 had a very similar role in mouse and human. And it's all, I've put the papers here so that you can look back at these contributions. So this was a great time. It was a lot of collaboration, a lot of interaction. I would stop my tennis games to talk to my friends about IL-10. It was just such an exciting time. And also the excitement was our collaborations and interaction with the lab members. So from IL-10, actually we went to IL-12 and back, and how pathogens and their products stimulate antigen-presenting cells to induce Th1 development, as I showed you in those summaries. And this was a collaboration. There's the young me in California, and set up this collaboration with Ken Murphy on the IL-12 Th1 development, together with Chi Song Shea, who was his student, and then Stephen McAtonia was the postdoc working with me on the IL-12 and the beginnings of IL-10, and David Fiorentino, who was then working with Tim Mossman when Tim left, came to work with me on the IL-10 effects on macrophages to inhibit the cytokines and antigen-presenting cell capacity. And the IL-10 was all made possible by the cloning of this cytokine by this person called Paulo Vieira, who actually is now my husband. So very nice collaborations and interactions during this time. These guys, Ken, David and Chi, these young guys are now professors. Paulo is a group leader at the Pasteur Institute, and Stephen McAtonia went in another direction, also successful, to import coffee to London, and Starbucks bought them, and they're like coffee millionaires. But enough about this. This was the interactive times and the talent of these young researchers and the fun we had in these interactions. So then the time came to come back to Europe, and I was recruited to the MRC National Institute for Medical Research at Mill Hill to interface research on immunology and infection, and try to get people who worked on infection and immunology to talk to each other, because this was often not the case. So this great National Institute for Medical Research at Mill Hill, which sadly is no more, this is now a flat space, I continued the molecular mechanisms, cytokine regulation and function in the immune response, and this continues to this day. But in 2006, I expanded the models of infection that were going in my lab and initiated research on the immune response in tuberculosis, because I thought working on this important interface, really maybe it was time that I worked on a pathogen that was devastating to man. So we now continue this work at the Francis Crick Institute, which the NIMR became part of, together with the LRI of Cancer Research UK. But what we first started on, I should preface with what was known in 2006 about the immune response to tuberculosis. It was known that MTB, an intracellular pathogen, primarily infects through the aerosol to the lung, and the infected cells can form a granuloma. I will not talk about granulomas today, but provided the activation of the right immune response takes place, dendritic cells will then go to the lymph node, activate CD4 T cells to produce interferon gamma and TNF. And actually this IL-12-driven interferon gamma production is shown to be fundamental by knockout mice from Joanne Flynn, Andrea Cooper many years ago, but also is important for control of mycobacteria infection in humans by Mendelian susceptibility mycobacterial diseases. So this Th1 axis is important in both mouse and man for resistance to the pathogen. Once these activated T cells are activated in the lymph node, they go back to the lung to control the pathogen. Another factor very much shown to be by Joanne Flynn in mouse knockouts to be important for control is TNF. And indeed, Joe Keane showed many years ago that actually it was also important in human, because when blockers of TNF, such as etanercept or infliximab, were administered to individuals with inflammatory diseases, those that had previously been exposed to TB recently could reactivate TB. So TNF and the IL-12 Th1 response, very important in mouse models and human disease. So, of course, I was coming with the thought of IL-10. And so we thought IL-10 has to be important. And yet there was a big debate as to whether it was. And we went on to show that indeed IL-10 was important for blocking macrophages from producing TNF, for blocking dendritic cells from producing IL-12, and for the Th1 development in the in vivo models. And it also blocked migration of activated Th1 cells from the lymph node back to the lung. But the effect was very small, and that's probably due to the fact that we're working with resistant mice, the C57 Black 6 mice, I'll tell you more about this, which the whole world was working on at that time. But I thought, this is the tip of the iceberg. How are we going to tackle this terrible disease, which is, apart from COVID recently, the leading cause of mortality from infectious diseases? And in fact, 1.5 million people still die per year. In 2020, that was the number, and it's going up as a result of COVID. So TB is still a very bad disease with 50% untreated mortality. The diagnosis is problematic. Drug treatment takes 6 months, and treatment monitoring with toxic drugs, treatment monitoring is problematic. And the BCG vaccine is variable, 0 to 80% efficacy, and it only works in kids to block TB meningitis. So a real problem. As an immunologist, what I found fascinating is that one person is infected with M. tuberculosis and proceeds to get the disease, whereas a quarter of the world has been estimated to be infected, exposed, but they remain asymptomatic. They could be subclinical and progress within any time. All you can say is that they were positive originally by the skin test that people used to do, but that wasn't specific for MTB. Now there's an interferon gamma release assay for MTB specific gamma interferon production in white blood cells that can be detected in humans who have been exposed. I'll go on to how later individuals may progress to TB later, but just to say 8 to 10 million cases per year, 8,000 cases in the UK in 2013, and in 2010, London was declared the TB capital of Europe. So at that time, I thought we've got to understand the immune response to MTB better to help maybe diagnosis and development of them, but also to capture the early infection and progression to disease and try and understand why. So it coincided with a visit to the Baylor Institute of Immunology, a research where Virginia Pasquale, Damien Chazabel, and Jacques Bonchereau were using blood transcriptomic signatures to look at inflammatory autoimmune diseases. And so I discussed with them. So our question was, why do some individuals remain latent healthy while others go on to develop active TB? What factors in the immune response may contribute to TB? And so the visit to Baylor made me think a global way to go, because I couldn't work at one cytokine at a time as I'd done for the basic research. We needed to get a capture of the disease. And so we asked the question, can human blood transcriptional signatures distinguish individuals with latent and active pulmonary tuberculosis? And this was a risk because Virginia and Jacques et al. Were doing this for autoimmune diseases, which may go to the show in the blood, but this is a lung disease. Could we see a blood signature? And I'm very happy to say that indeed we described an interferon-inducible neutrophil-driven blood transcriptional signature in the blood of human active tuberculosis patients that was missing in the latent individuals. And I'll take you through this of our work published on this. So we published this blood signature of genes that were upregulated or downregulated, seen in the active TB, but missing in the healthy and the majority of latent. I'll come back to that. We also found that the signature was diminished upon successful treatment. And this original work was led by Matthew Berry, a clinician in the lab, then Chloe Bloom continued, and she showed that this treatment was variable according to the different people. And more recently, we published a collaborative paper with our Leicester clinical collaborators in JXMed, that the transcriptional blood signature reveals different responses after treatment in clinically defined subgroups. And all I want to show you, this is a modular response, which I'll talk about more later, but you can see this interferon signature was diminished in standard treatment, took longer in extended treatment. And all I want to say is that in these TB drug-resistant individuals, you really couldn't see the signature going down, and therefore the signature was telling you who was responding to treatment or not. Now, we looked in detail at the signature, and I'll go on to more data showing that it was consisting of interferon gamma-inducible genes, which we expected to be, and type 1 interferon-inducible genes. But just to break off and say what's happening at the clinical level, that reduced blood signatures obviously would be important to support TB diagnosis and treatment monitoring, which is normally sputum clearance, which takes 2 months. And so these are being developed by a lot of people around the world based on these original findings. And we have various papers contributing to this and studies funded by BioAster, Biomeria, who's developing this, and the university hospitals in Latency, to see, can we see this early TB and help to get in sooner and be able to start treatment, and can we determine whether people are responding to treatment or not. But that's the major part of the clinical development that I'll talk about there. Back to the signature as an immunologist, we wanted to know what's there. And we did the signature in London, where I showed you the signature there in the active TB patients. We recapitulated it in South Africa with Robert and Kathleen Wilkinson and the team. And you could see that the signature was here in active TB, but missing in the latent. Everybody was latent in South Africa. They'd all been exposed. And so that was what we published. And then we looked further, again, using this modular approach to show this was actually reproducible in a training set in London, then another set in UK, and then in a validation set in South Africa. And these are clusters of genes that are expressed together against the controls. So this, for example, was an interferon-inducible module consisting of lots of interferon-inducible genes. These one were myeloid genes turned on here. And conversely, in group A strep and staphylococcal infections, which can sometimes be confounding, you could not see much of this interferon signature, but you could see a plasma gene signature that was different in the blood of active TB patients. Now, Virginia Pasquale basically had this adult SLE signature with the plasma gene and the interferon-inducible genes. And we thought it has to be interferon gamma-inducible genes because this is TB. And Virginia said, no, it must be type 1 interferon-inducible because in SLE, this is what we see. And look, the signature looks very similar for the interferon-inducible. So Matthew Berry looked at great detail at these genes that were turned on, and in fact, found that it was type 1 interferon-inducible genes dominating this signature. And we've recapitulated that since in Leicester, South Africa, and all the cohorts. And you could see that actually it's not interferon gamma and interferon alpha beta, but it's genes downstream of these cytokines that were turned on, and often dominated in the active TB patients by the type 1 interferon-inducible gene signature. And this correlated with the extent of lung radiographic disease and was diminished on treatment, therefore suggesting a role in pathogenesis. So we could see these interferon-inducible genes in the whole blood. And then Chris Graham in the lab separated the neutrophils, monocyte CD4 and CD8, and you could see genes induced in the neutrophils. Some of these genes in the monocyte, but not in the T cells or B cells. So why were we excited? Because type 1 interferon and TB had really not even been thought about. We always thought about it in the context of viruses and cancer, but in Listeria, it had been shown by Emily Nano and others in the past to exacerbate bacterial infection. So what was going on in TB and why had it been missed? Why had type 1 interferon role in MTB infection been overlooked? So we looked at papers, and this was now 2010 when we published our paper. And we found 2 studies of Gila Kaplan where she'd used hypervirulent strain of MTB called HN878, which had got isolated from the clinic. And this was associated with high levels of type 1 interferon upon infection of mice and decreased Th1 and shorter survival time. In addition, she wasn't using the B6 straight mice, but a F1 cross. Then she published another paper in 2005 with her group where they had interferon receptor knockout mice, again on a different background, and here with the clinical isolate of TB. And there she showed that these had longer survival times. So there was some data there, but it had been kind of left to the side. So this human data in TB, type 1 interferon signature, triggered quite a lot of activity. Alan Scher's lab, thinking that poly-IC may protect against MTB infection, actually showed that it caused a 2-log-fold increase in lung MTB bacterial load, but this was not happening in interferon alpha-beta receptor knockout. And then in our lab, we thought, okay, maybe we have to elevate the type 1 interferon in the B6 mouse. And so co-infection with influenza with Andreas Vark, Alan Scher, and Paul Redford leading this study showed that mycobacterial co-infection with flu exacerbated TB through a type 1 interferon receptor dependent pathway. And then Finn McNabb and John Eubank in the lab went to show that if you blocked the TPL2 pathway, which blocks type 1 interferon signaling, then you could protect against TB, and the TPL2 ERK signaling promoted host resistance by getting rid of type 1 interferon signaling, and on a type 1 interferon receptor background, this didn't happen. So elevated levels of this type 1 interferon was really important, which could be brought about maybe by the genetic background of the MTB strain, genetic background of the host, and this provided a late sustained high dose of type 1 interferon ensued. This would then block IL-12 and TNF production and also induce IL-10 and also block the gamma interferon activation of macrophages to control MTB infection and produce these cytokines. And these were papers from our own lab and also from Katmeyer Barber and others through the years with Alan Scher. And what I should say that following our findings and their poly-IC data on exacerbation of TB, Katmeyer Barber and Alan Scher went on to publish a seminal paper showing that host-directed therapy of tuberculosis was actually based on the IL-1 and type 1 interferon crosstalk. So protection by IL-1, IL-12, TNF could be abrogated by interferon gamma, type 1 interferon, which really blocked all these pathways. So at that stage, we thought, okay, exacerbation through poly-IC, co-infection with flu or genetic mutations may lead to high levels of type 1 interferon, but could we find a tractable mouse TB model that resembled human TB? Because we really were working on tuberculosis to try and relate human findings to mouse models and mouse models to human disease. And this took us back to the work of, seminal work of Igor Kramnik, first with Barry Bloom, showing genetic control of resistant to experimental infection with virulent MTB tuberculosis. Actually, that virulent was Erdmann in those days, for those who know TB. And what they showed was that in the lung of C3H FEJ mice, MTB, these mice were susceptible to TB within a few weeks of infection, whereas the MTB resistant mouse was the CTG7 Black 6 mouse. And they went on to describe a new locus termed susceptibility to tuberculosis 1, which I'm not going to go into the genetics, designated SST, when this was mapped, and the lung lesions in these congenic mice were characterized by extensive necrosis and unrestricted extracellular multiplication of MTB. And you've heard Russell Vance speak about the later collaboration where then this susceptible locus was found to be mediated by R1 receptor and driving type 1 interferon dependent susceptibility. But what I'd just like to go back to is that Igor Kramnik has published some very nice reviews and papers on mouse models of human TB pathology, roles in the analysis of necrosis, and development of host-directed therapies. And his line is, thus far, the mouse model of necrotic TB lesions in C3H-EB FEJ mice displays pathomorphological diversity of TB lesions with similarity to necrotic lesions in humans. So we thought, okay, this is the mouse we want to work on because it'll have the diversity of genetics that may be equivalent to that seen in humans. And can we then see whether the blood signature resembles that of human TB patients? So this time, using a modular approach based on WGCNA, we found in the London cohort, the South Africa cohort, and a more recent Leicester cohort that the type 1 interferon inducible signature was here indeed in the human blood, and 2 type 1 interferon inducible signatures, and also a neutrophil inflammasome and innate immunity neutrophil-driven signature. And so we looked at the blood of mice, which were either B6 mice or C3H-EB FEJ mice, infected with a low or high dose of either H37RV, the lab strain of M. tuberculosis, or that clinical isolate that I've referred to earlier, HN878. So we varied the Mycobacterium tuberculosis strain, the mouse genetic background, and the dose of MTB. And what I think you can see very clearly is that this signature of human TB of type 1 interferon inducible genes comes up with a high dose B6 in the HN878, but there in the susceptible mice with the lab strain or the clinical virulent strain, again, here also with the other type 1 interferon signature. And again, the same was true for this inflammasome, neutrophil signature, innate mediators, and this neutrophil-driven signature here. So this was really amazing that we had a whole blood signature of certain mice that resembled human TB. So we went to look to see the role of type 1 interferon, and this was work from Lucia Moreira Teixeira, some of which has been published in susceptible mice and other unpublished, so please don't tweet. And here the low-dose TB in the B6 mice, MTB, showed no effects of type 1 interferon receptor knockout mutation, whereas with a high dose, you could actually see increased bacterial load, and this was much reduced in the IFNA knockout. And in the C3H-EB FEJ mice, which are the susceptible mice described by Kramnik, showing the blood signature, you could also see that this bacterial load was diminished by treatment with anti-IFNA monoclonal antibodies. So there's a lot going on, but type 1 interferon seems to have a very dominant role in exacerbating the extreme levels of tuberculosis. So what is it doing? So Lucia Moreira Teixeira showed with Vangelis Stavropoulos and other collaborators that in fact, when you infected these HN878 strain into the C3H-FEB FEJ mouse, and then blocked or not with type 1 interferon receptor antibodies, you could see a reduction in the bacterial load, a reduction in the lesion burden, reduction in the necrosis in the lung, and a reduction in the intra-alveolar necrotic debris. And most exciting was this reduction in the type 1 interferon-induced neutrophil extracellular trap formation, which is seen by this staining with citrulated H3 antibodies and MPO for the neutrophils. And you can see the neutrophils are reduced, but they're still there, but the anti-IFNA really completely abrogates the net formation as we showed with other collaborators advising us. So this was exciting. So what about the lungs of these resistant and susceptible mice? Could we find something relevant to the resistance? And what's striking was that in the B6 mouse with the H37RB strain, you could see an immunoglobulin signature here and TB B-cell NK effector signature also in the resistant mice, which then disappeared in the lungs of the susceptible mice. Of course, the IFNA is interferon is dominant, but going along with this reduced protective signature in the susceptible mice, you see myeloid granulocyte IL-17 response. And so this showed converse responses in the lungs, in the airways of resistant versus susceptible mice. And we could confirm this by immunostaining, which you can see the CD3 is the red, the beta-20 is the B cells, which you can see in the resistant strain and even the susceptible strain with a lab strain of MTB. But as soon as you got to the virulent MTB isolate, with high dose in the B6 mice, this was completely gone, as well as in the susceptible mice. And one doesn't know is if they couldn't make the B and T cell response or whether the lung deteriorated to the extent that you just really can't develop this. So to summarize that part of the talk, type 1 interferon, we've shown activates neutrophils and induces netosis in susceptible C3H-FEBJ mouse. We're still asking the question, does type 1 interferon drive recruitment of permissive monocytes and neutrophils in susceptible mice from the bone marrow? Does this contribute to the pathogenesis, or is it really only in the lung? And we and Katmaya Baba and others have shown that type 1 interferon will reduce IL-1, IL-12, and contribute to IL-10. But what we're still asking is, in the resistant strain of mice, do low amounts of type 1 interferon induce protective effects? Because we know that type 1 interferon can actually activate NK cells and T effector cells. So there's still quite a lot to find out. And we're continuing that, as I'll show you, for our future plans. But just for the final part of my talk, I want to go back to the problem of tuberculosis and latency. And there's a big debate about what is going on. So one person will progress to TB, and it was thought that anybody could, who was latent and asymptomatic, but positive for these skin tests and the interferon gamma release assay, could reactivate at any time in their lifetime. So this really requires a lot of longitudinal studies. And what we could say about latent individuals is that of the 5 to 10% of who progress, which are these? So TB was shown by PET CT scan by Cliff Barry, Rob Wilkinson, Douglas Young in 2009 to be very heterogeneous. The individuals could have cleared the infection. They could have controlled persistent infection. They may have subclinical active TB, or they could be false positive, as we found of some in the clinic. So all you could say is that we're positive for the skin test, positive for the IGRA test, but you can't distinguish active TB from latent, which we now could with our blood signature, but you couldn't determine which will develop active TB from those latent individuals. So this was our original signature that we published in 2010, Berry et al. Paper, where this was the active TB, and for the most part, all the latent individuals shown in black were missing a signature. But you can see here there were a few of them clustering with the active TB. And if you take away the active TB, you see these later individuals really had the signature of active TB. And if you use the modular for these outliers versus latent TB, you could see that the LTB outliers were starting to get this interferon-inducible signature of type 1 interferon, such as like active TB. But this was a static 1-point signature. So we could see it was heterogeneous, latency, but how could we get at this further? So what we really needed to do is to do a longitudinal study of contacts of TB patients that were latent and see when they would progress to active TB. So as I said, to date, the literature stated that reactivation of latent TB can occur at any stage over a lifetime, but could transcriptomics tell us anything about TB progression? So we collaborated with Pranab Halder and colleagues at the University of Leicester, which was an important place to do this study because it's a low TB burden setting which minimizes the risk of MTB reinfection that can happen in high burden countries like Africa. So here we had patients that had been diagnosed with active TB, and then their contacts were recruited and they were followed up over 3 years. So some of these contacts remained IGRA negative and healthy over these 3 years, and they had a transient low blood signature in these individuals that we followed. There were IGRA positive contacts who actually got a transient signature that remained, that then went down. And then we had in this early pilot studies, 9 who progressed to active TB, which had a persistent high blood signature in 70% of them. So we wanted to look at this in more detail and really understand what happens when people get exposed to Mycobacterium tuberculosis. So we increased the number of household contacts recruited who progressed to TB. And what we could do then is link the clinical data from the patients with our colleagues in Leicester and the M. tuberculosis genome data to ensure that the people who progressed to TB had the MTB strain of the people who they'd been in contact with, and it was not reinfection. And then we could analyze their signature. And so I just want to show you this very clearly because it's really quite important. The TB patients that were diagnosed, their MTB genome was sequenced, and TB diagnosis could take some time for various reasons, because you have to be able to grow the bug and you can't always grow it. But in that time, 356 household contacts were sampled and clinically monitored over 3 years. And of those 356 contacts, 20 contacts only progressed to TB, and those progressed within 2 to 3 years. And the MTB sequence was obtained from these contacts and verified to be the same one from the people that they'd been exposed to. Now, from the clinic, the progressor latent or LTBI state has been characterized into 2 phenotypes. So there are people who have no symptoms, working backwards from those who progressed. And there can be these incipient TB patients who have no clinical symptom, no radiographic abnormalities, no microbiological evidence or anything. You could have subclinical TB, which have no symptoms, but they can have radiological changes. But this could be lung cancer or other kinds of pneumonia. Or conversely, they could have microbiological evidence of TB, but no changes in their lung. And this is always a worry that it could be a contaminant or a historical strain. And then there were the clinical TB patients who were contacts, who basically had radiological features and microbiological evidence. You could grow the MTB and sequence it. And what's interesting is the time before diagnosis of these progressors in our study. So the clinical TB that progressed very quickly was in less than 20 days, and they have a signature of TB, but it's much reduced compared to an active full-blown signature. The subclinical progressed within 20 to 41 days, and the incipient progressed between 41 to 125 days, and sometimes within a year or two, but on average. So what we did was look at the first the gene expression in these individuals, and actually in the incipient, you could see low numbers of genes that were up or down regulated, only 94 genes. This progressed within the subclinical TB and then was raised much higher in the clinical TB with 572 genes upregulated. But still, this is lower than in fully blown active TB. And what we could see then using the modular approach, that the subclinical disease had interferon-inducible genes coming up and then reminiscent of TB. We could detect genes by the volcano plot I showed you earlier, but using the modular compared to the control with incipient, we couldn't detect genes going up, but what we could see was NK and T cell signature going down. And if you remember, that is the protective signature in the resistant mice. that we talk about. Going back, we have selected a 30-gene signature of these asymptomatic individuals, which is then being tested to see if it can diagnose TB earlier by clinicians and bioMérieux and others. But what I should say about this signature is that this blood transcriptional changes are actually changes that are becoming more pronounced with disease progression. And Will Branchett looked at the analysis together with Probia Chakravarti, showing common pathways enriched in these upregulated genes in all three clinical groups, with type 1 interferon stimulated genes being highly enriched and showing this increase as Tabon showed in the volcano plot analysis. So it looks like you're getting TB progression that is happening within 1 to 2 to 3 years. And this would fit with most progression happening early and a small number only reactivating later in their lives, as now published by other groups looking at background epidemiology. So where does this leave us? Looking at further studies in our active TB patients and their recent contacts to really try and understand at an immunological and molecular level what is happening in the airways of these individuals that have subclinical disease or are incipient, that will eventually progress to active TB. And so the airway samples are taken at the first visit and at 3 months, and also from these latent individuals that will remain healthy over 2 to 3 years and during their life. And these have controlled MTB infection. And again, we get the airway sample for the first and third 3-month visit, and they're being monitored over the years with a blood signature and at the clinical level. And so we will be able to relate the clinical phenotype to what's happening in their airways. And Will Branchett in the lab is leading the team to work on the at the molecular level, flow cytometry for immunology, RNA-seq, single-cell RNA-seq, and ATAC-seq to try and understand the events that determine whether one person goes on to TB, whereas the other remain healthy. And in parallel to this human study, which is quite challenging, we have studies going on in the susceptible and the resistant mice, where we're looking at the host airway samples and really relating the molecular data we obtain in the airways of these mice to that we obtain in the human data, to ensure that any targets that we're following really match with human TB. And this is, of course, underpinned by the research program of cytokine gene regulation in my lab, and by a Wellcome Investigator Award and a Lagaisha Foundation. But to finish, I just want to acknowledge, this is just the TB work I'm acknowledging through the years. I acknowledge the basic research first. So the human TB work was started by this clinician, Matthew Berry, who was actually resident in my lab and collaborating with Onmin Khan in the clinic, as the clinical work in London at St. Mary's Hospital, then Hillingdon. So we thank the patients and the healthy volunteers. And this has been a journey with other physicians, Chloe Bloom, Simon Blackley, and Christine Graham, supporting all the RNA work in the human studies and in the mouse mechanism studies, where this was started with Paul Redford, who was my student, and then a postdoc investigator scientist. He's now at GSK, all underpinned by Vangelis Stavropoulos, expert on MTB mouse models, and then some of the mechanisms Finn McNabb through the years, and then bioinformaticians in the lab, Akul Singhania, phenomenal, and Olivia Tabon in the more recent work, and then mechanisms of MTB and type 1 interferon, induction of IL-10 and pathology. Lucia Moreira Teixeira, along with these guys, did a lot of this work, which is now being led by Will Branchett. And we have to thank all our science technology platforms at the CRIC, for which we're very, very fortunate, including our pathologists, collaborators through the years, Andreas Bach, Katmea Barber, Margarida Saraiva, who's actually an ex-postdoc of mine, who now has her own independent group in Porto. She continues work on IL-10, the basic, and then we collaborate on certain TB work, which has expanded out to other hospitals in Cape Town, Royal Free, and lots of funding through the years, because at the beginning, I was accused of being a mouse immunologist who would not be able to do human TB research. So, you know, hang in there, young people, when you get these negative blows. But these are our Leicester clinical collaborators. We thank all our past collaborators, and then our present lab. So this is from mouse models in TB research to human TB research and back. And so Will is leading this, supported by Ala Aldibouni, who's an expert histologist looking at the airways, and Vangelis Stavropoulos, who has really done all the MTB growing in our lab and MTB mouse models, and teaches and collaborates with the team. And with Margarida Saraiva's lab on a La Caixa Foundation, we collaborate on the mouse models. We're now recruiting a bioinformatician, and we're indebted to our clinical collaborators whom we meet with every week. And just sending those samples down just from Leicester has been a challenge. We had to work out the couriers over a couple of months to ensure that these precious samples obtained by Pranab Halder, the lead clinician, Raman Verma, and Joe Lee, who has been a long-term collaborator on our previous studies, and now Kim Ji Wang, clinician, and Ismail Navaska really supporting at the administrative level these clinical, difficult, challenging studies, conducting PET-CT scan that then can be brought together with our molecular transcriptomic and RNA-seq data of the human airways. I'd just like to thank bioinformaticians who've helped us along the way and still help us. Akul has left our lab, but he still collaborates. Probia Chakravarti supports us during these transitions as people leave. But what I'd also like to say is that I'm still an immunologist studying TB, and this is underpinned by the basic immunoregulation focus in my lab, which is IL-10 gene regulation through the years, of which we've published a lot. But we've really decided we've got to go in vivo on this, and this has been led us to the gut and the peritoneal cavity. And Luke Cox was continuing with Leona Gabrisova on IL-10 gene regulation with MAF and BLIMP. And then this led us to the gut, where we had some very nice findings that are soon to be published in collaboration with Fiona Parry and Claire Pearson, and Marisol, Will, Ala, everybody, and Shumei have all contributed to this gut work, which now we're happy that Hannah and Kunyan will be continuing in full force in the future. When Kunyan joins, Hannah's just joined in September, together with Shumei and our collaborators. And Marisol underpins this with beautiful work in the peritoneal cavity, migration of neutrophils and monocytes to the peritoneal cavity, and a lot of that underpinned by type 1 interferon in this project and the human TB. Wait and see where the work is leading us in the gut, and look out for our publications. With that, I leave you and thank you for your attention, and look forward to any Twitter questions. Okay. Thanks very much. Bye-bye.
Thank you so much. And that was fabulous. Thanks for sharing your efforts and those of your colleagues in trying to understand the immune response to TB. And it's very exciting to see how you can identify this predicted signature in the subclinical setting. Hopefully you're able to translate that. And I hope that will have a tremendous impact in trying to treat this devastating disease. So for those of you that are still with us, or if you're listening in YouTube, you have an opportunity to ask Anne questions. I want to remind you, you can go to our Global IMMUNOTALK Twitter account, and you will see there's already there a tweet that says, ask questions for Dr. Anne O'Gara here. You just have to very easily reply, just click on that tweet and include your question and just add the hashtag Global Immuno. I would like to remind you that Anne will use the Global Immuno Speaker account, so also include the @ImmunoSpeaker. Thank you so much, Anne. It was really very special to connect with you and to listen to this very important work. Thank you.
Thank you.
Okay, bye everybody. See you next week. Join us next week for Dr. Howell's talk. Bye-bye.